The seedling root screen is a high-throughput, controlled environment system for the study of seedling root architecture. Measuring root traits at early growth stages can be important for looking at differences in seedling vigour and differences between genotypes, as well as responses to diseases, seed dressings, nutrients, or other treatments. Step 1. The seedling root screen system. The seedling root screen is a hydroponic system consisting of a tank with reservoirs for the hydroponic nutrient solution. The separate trays allow for blocking of treatments in solution. Perspex rods slot in above the reservoirs that hold the blotting paper, which the seedlings are placed onto. The bottom of the papers sit in the nutrient solution, which rises up the papers through capillary action. This setup was designed by the University of Nottingham in the UK. In this example, each tank can hold 200 seedlings. Step 2. Seed sterilisation and germination. The seeds are surface sterilised before being pre-germinated. This helps to reduce bacterial and fungus growth on the seeds and the papers whilst they're in the root screen. Surface sterilised seeds are placed on moist germination paper in Petri dishes to pre-germinate. Step 3. Pouch setup. Before the pre-germinated seeds are placed onto the blotting papers and into the root screen, the pouches are set up and pre-wetted with the nutrient solution. To set up the pouch, a black plastic sheet is placed on a clean bench, before the blotting paper is placed on top and covered with a second plastic sheet. 2 pouches can be attached to one rod using the butterfly clips. These are then placed within the dividers in the tank holding the nutrient solution. The papers are left to soak up the nutrient solution until completely wetted, which can take around 4 hours. Step 4. Selecting germinated seeds and placing them on the papers. Seeds are selected for transfer which have similar stages of growth. This is judged based on the length of the emerged radicle and the seed's appearance. They are moved using sterilised tweezers and placed on the blotting paper, immediately below the plastic rod with the root facing down. The plastic sheet is gently wiped down to ensure it adheres to the paper. Step 5. Removing the pouch, checking root growth, and taking photographs. At the end of the experiment, when the roots are reaching the edge of the blotting paper, the root systems are photographed. Photos can be taken at any point between setup and ending the experiment to measure root development. When taking photos, it is important to keep all settings on the camera the same and to position the papers in the same position and the same orientation to help with image processing. Taking an image of a ruler at this stage allows pixels to be converted to centimetres during image analysis. Step 6. Image analysis. The photographs are renamed with treatments and replicate numbers and cropped before being analysed using RootNav1 software. First, identify the root origin, which is the point where it emerges from the seed. Then add in the primary root ends. Click Analyse and adjust the root paths as required. Next, move on to the laterals, adding tips. Click Analyse and adjust the root paths. When an accurate representation of the root system has been achieved, click Measure. RootNav will then calculate a range of root parameters including length, tip angle, tortuosity and convex hull. These can all be statistically analysed. The program can also overlay the individual root systems within a treatment to create a heat map, and these can be a nice visual support for the findings of the data. We hope you enjoyed this instructional video on the seedling root screen. Please do get in touch if you want to know more or discuss root phenotyping. Keep up to date on the Root to Res project by following us on social media or visiting the website.